2.3 Carbohydrates and Lipids. Concepts to know are the features of carbohydrates. Carbohydrates are organic molecules composed of one or more monosaccharides. They include sugars and polysaccharides. Sugars include monosaccharides and disaccharides. They are sweet, saccharides and sugars. They are small and soluble sugars. Sugar is more important energy form in many organisms. Two monosaccharides can be made into one disaccharide. Polysaccharide is insoluble and complex and does not taste sweet. You need to know the word equations and be able to tell if a word equation is a representative of condensation or hydrolysis. In this case, all three equations are representative of condensation because they join molecules with the release of water, whereas hydrolysis is a split of molecules using water. You need to know which part of the condensation hydrolysis reaction is in a diagram. For example, here is a glycosidic bond between carbon 1 and carbon 4. You need to know the difference between the structure of amylose and amylopectin. Starch is made up of polysaccharides, amylose and amylopectin. Amylose chain or alpha-glucose monomers joined by glycosidic bonds are unbranched. A long chain of glucose monomers form a helix. A long chain of amylopectin, a chain of alpha-glucose monomers, are joined by two glycosidic bonds, 1 and 4 carbon glycosidic bond and a 1 and 6 glycosidic bond. The second bond causes it to be branched. So right here would be a 1 to 6 carbon glycosidic bond, and that would be 1 to 6 as well. You need to know the relationship between structure and function of starch. The function of starch is to store glucose. It is a large molecule that is insoluble. Starch does not move water into cells, so there is no osmotic effect. There is a helix structure and is compact to make storage easier. It is branched structure of amylopectin means there are many ends, which allows starch to be rapidly broken down. The structure of glycogen and relation to its function. It is similar to amylopectin, but there are more 1 to 6 glycosidic bonds, meaning there are more branches. Large and compact, good for storage of glucose in muscle and liver cells. It is insoluble, so there is no osmotic effect. More branches means that it can be broken down faster for cell respiration. The structure of cellulose versus its function. It is long and straight molecule, and the molecules are very close together. This allows hydrogen bonds to form between molecules. This associates to form microfibrils, great for strength to form a cell wall. You need to be able to compare the structure of starch, glycogen, and glucose and cellulose. For amylose, there are no branches and a helix structure. For amylopectin, there is a branches because it's a 1 to 6 glycosidic bond. For glycogen, it is similar to amylopectin, but there are more branches because there are more 1 to 6 glycosidic bonds. For cellulose, there are no branches and creates a straight molecule. Alternate flipping of glucose monomers in the polymer. You need to be able to calculate the body mass index with the formula mass in kilograms divided by the height in meters squared. The main types of lipids come in triglycerides that are composed of 1 glycerol and 3 fatty acids. Examples are fats, which is a solid, and oil, which is a liquid. These are hydrophobic. The phospholipid is like the triglyceride, but one fatty acid is replaced with a phosphate group. This is amphipathic, meaning this is hydrophilic and this is hydrophobic. It is found in the phospholipid bilayer. Lastly, a steroid. This contains 4 carbon rings, 3 that are hexagonal and 1 that is pentagonal. This includes cholesterol and male and female sex hormones. Lastly, there is the saturation of fatty acids. There is saturated, monosaturated, and polysaturated. There is also unsaturated, as well as cis and trans unsaturated. Cis is hydrogen atoms bonded to carbon sharing the double bond and are on the same side. Trans is a hydrogen atom bonded to carbon sharing double bond on opposite sides. You also need to be able to determine the omega number for fatty acids and understand that 3 covalent bonds form between glycerol and fatty acid. You have to be able to draw the structure and of the saturated fatty acid. You need to know the advantages of lipids versus carbs in long-term stores of energy. Carbohydrates stored in liver and muscle cells are the main energy source, but lipids or triglycerides are stored in adipose tissue, release twice the amount of energy per gram. They both have equivalent storage space, but carbohydrate is the main energy source and lipids are the most energy source.